everyone, and welcome to another video on enzymes. This is chapter 3.2, Mechanism of Enzymatic Reaction. In this lecture, we'll be learning about two different enzymes today, Catalase and Amylase. Enzymes often end with the word ASE, ASE. So yeah, that is a pro tip for you. Anyways, let's start with just a little introduction on what is a rate of reaction. So rate of reaction is basically the speed of conversion of substrate into product. That makes sense, right? Substrate, if you convert to product, that is a reaction. So the speed of which that happens is rate. Rate is always has to do with time, so it's always per unit time. For example, per second, per minute, cm cubed per hour, something like that. It always has to do with time. Now, how to measure rate of reaction in an experiment? Okay, so to measure the speed of conversion of substrate to product, right, we need to at least measure something, right? So we probably would measure rate of product formation, so focusing on the product, yeah, or we measure the rate of substrate disappearing. So if the substrate finishes, like it diminishes over time and becomes zero, we know that the reaction is complete. So what are steps to get the rate of reaction exactly? So to get the rate, we need to, number one, measure product formation or substrate disappearance in some form or way, whether it's concentration, volume, so on and so forth. So measure that, and we plot a time cost graph. The time cost graph is basically a graph where the x-axis is time. So we are charting what happens to the reaction over time. Now the y-axis here could be product concentration. Okay, the square brackets mean concentration. The y-axis here can also be, substrate concentration, or even, enzyme substrate complex concentration. But that's more rare. The one that is more common is product concentration or substrate concentration. But it's called a time cost graph again because the x-axis is time. Now, after we plot this time cost graph, then we can find the gradient. If we find the gradient, okay, this means we are dividing some concentration by time. This would give us the rate. So yeah, let's talk about rate of reaction and the time cost graph using the two enzymes we'll be learning about today. Starting with catalase. Hydrogen peroxide is a toxic metabolic product in tissues, and therefore all living organisms, most living organisms, have catalase enzymes in order to break down this toxic product into harmless oxygen and water. And this can be found in potato extract, in liver extract. Okay, so we buy a liver from the market, we blend it, or yeast extract. So all of these organisms, again, most living organisms, have catalase and can convert hydrogen peroxide into oxygen and water. Now, how do we measure the rate of reaction? Now, in this case, because hydrogen peroxide is mostly liquid, okay, it's very easily oxidized, but usually liquid. Oxygen is gas, whereas water is liquid. Because of the different states of matter, we can measure the rate of oxygen release by collecting gas or bubbles. Yeah, so this is the normal setup. This is for gas collection. So we have the reaction mixture here. Let's zoom in a little bit. How do you zoom in? Yeah, right. So if you see here, we have a reaction mixture at the bottom here, catalase with H2O2. Okay, and then you have a thermometer maybe to keep the temperature constant, and whatever gas that's produced will enter this delivery tube, go inside here and displace the water here. So over time, you can see how much oxygen is produced. So yeah, that's a way to do it. Or as I said just now, you can count bubbles. So obviously counting bubbles is not that accurate, but it is a way to do so. And hopefully when you get to do labs, you would be able to do the experiment as well of counting bubbles. Okay, anyways, if you do the experiment I just described down below here, we would have collected oxygen, and this would be the graph I would chart. So the volume of oxygen produced over time will look like this. So at first nothing, and then it increases rapidly, okay, and then it starts to level off. Now what are we plotting here? On the y-axis is actually the product concentration, because oxygen is our product, right? So this is product concentration against time, making this a time course graph. So that's volume of oxygen. How about speed? Can we talk about speed for a while? So basically the volume of oxygen is produced very fast, and then it kind of slows down here. It's still produced, but it's slowing down. And then at around 25 cm cubed, it kind of stops. Now this tells us that the rate of oxygen release is high at first, and then decreases, and then goes to 0. When the graph plateaus, the rate equals to 0. The reaction has completed. Now just a reminder that the rate equals to gradient. So if we do want to be OCD about rate, I mean, I'm kidding, but like we do want to calculate the rate at every single minute, for example, every single second in this case, then we can take a ruler and we can draw a line that is tangent to the curve like this. And we measure the gradient of this line. Okay, and we can do this for every, say, 20 seconds, for example. We can do this another time, another time, and another time. Yeah, and you realize that the steepness of this line go from very steep, which means the gradient, the number that you calculate would be very high, all right? And this one would be telling you also that the rate is very high. Here, less steep, so rate is still high, but is not, sorry, rate is lower now, but it's still increasing, it's still going on. Until the line becomes horizontal, gradient equals, oh, I did a very bad job at a horizontal line. Horizontal line, gradient equals 0, and therefore rate equals to 0. So yeah, that's a reminder for you. Gradient of a horizontal line is 0. So when the graph plateaus, rate equals 0 in a time course graph. Now, sometimes the exam would ask you to calculate the initial rate of reaction. How would that work then? Now, initial rate of reaction is calculated by drawing a line as close as possible to the starting point and close as possible to the line from the starting point. Yeah, so the gradient of the curve at 0 seconds or 0 time in whatever units. And this would give you the initial rate of reaction. So just what the rate of reaction is at the beginning of the reaction. So that's the initial rate of reaction. Okay, so we're done with catalase. Let's talk about the next reaction, amylase. So amylase converts starch into maltose. Now there is a problem here. Both starch and maltose are liquid. This is liquid. This is also liquid. They are both colorless. So how can you measure something that looks exactly the same? Now, we know a test though that works on starch and not maltose. And yep, you're right, it's called the iodine test. So starch can be tested for by iodine tests, and we can use iodine tests to find out the rate that starch disappears. So yeah, what do? We take a reaction mixture. So imagine we are doing the experiment with amylase and like starch in a beaker. every, like, say 1 minute, we take a drop, just a tiny drop, and we test it with iodine solution. The color would change from yellow to dark blue, telling you that there is a high concentration of starch at first. But over time, as you test every single minute, you realize that that dark blue color kind of becomes like a dark brown, and then later on a dark yellow, and then yellow. And this tells you that starch is completely hydrolyzed and converted into maltose already. So basically, in the beginning of a reaction, starch is present, such concentration is high, whereas at the end of the reaction, there is no more starch. Starch concentration equals to 0, leading to a negative iodine test result. Now we can use this spotting tile that we can see here that I just described with, and this is kind of a qualitative kind of way. So it's not very accurate, it's just by your eyesight, right? And how you differentiate colors. But there is a quantitative method using a colorimeter to measure the color intensity. So this is a colorimeter, this is a fancy one. Our lab has an older version, quite bulky, quite chunky, but it also uses this what we call cuvettes. This is how a colorimeter works. So this is a cuvette. The colorimeter actually measures light absorbance, okay, for liquid in a cuvette. Cuvette, not curvette, cuvette. We fill some liquid in here, so this would be like a few drops of the reaction mixture plus a few drops of iodine. And what it does, what the colorimeter does, is that it shines a ray of light of a set wavelength through the liquid, and the sensor will detect how much light has passed through. So you can imagine if it's dark blue, a lot of starch is present, and therefore it's very dark in color, right? So less light can pass through, and absorbance is high, relative to water, of course. Now, if at the end of the reaction there is no more starch present, it will be a light yellow because the iodine test will be negative, right? And therefore more light can pass through the liquid, and therefore absorbance is low. Now, as I said just now, bear in mind that this is relative to water. Distilled water is set as a control. This is set as 0 arbitrary units. So all of these light absorbances are measured relative to this water, and therefore the units are in AU, arbitrary units. It's not real unit, it's like relative unit. Okay. So yeah, if we do that, we get a lot of results, and they are quantitative. So there's actual values that comes up. Right. We can plot a graph of light absorbance in arbitrary units against time in seconds. Now, light absorbance here is really talking about our substrate concentration, because as we said, at this point where the solution is dark blue in color because of the starch present, light absorbance by the liquid is high. Less light passes through. So it's high at first, but later on is low because it's yellow, and starch because starch is absent, and the reaction is completed. So what can we say about the concentration of starch, which is the concentration of substrate? It increases and then levels off. The speed of starch being used is very fast. Okay, very fast at first. It's like a rapid decrease, and then it kind of slows down, and then it stops. The rate of reaction is high. Look at the line. If you draw the gradient, the tangent to the curve, and look for the gradient, this is pretty steep. This is less steep, and this becomes almost 0, right? So it goes from very high to decrease rate of reaction to 0. So what actually is going on? Okay, what happens during the course of the reaction? Now to illustrate my next slide, we're going to use this kind of diagram. This here is an enzyme, this here is a substrate. But you know what? It's kind of like hard to draw. So let's just draw a Pac-Man, and this is supposed to symbolize an enzyme. And this pizza piece, this pizza slice, is supposed to show our substrate. And our half pizza slices are our products. Okay, this is just easier to draw for me in the slides. So yeah, this is my cute Pac-Man enzyme. Maybe let's call it Catalase, because you know, enzyme. Anyways, yeah, so what happens during the course of reaction to cause the rate to increase, to be high, and then decrease and go to 0? Right, what's going on there? Now, when rate was high at first, in the beginning, right, of the reaction, there is high substrate concentration. The substrates are in excess, so you have only so many enzymes. There's more substrates than enzymes, right? So the reaction rate is high. However, over time, more substrates get used up, okay? And over time, lesser substrates are available. So a lower substrate concentration occurs, right? And after all those substrates are kind of like converted into products, basically there's no more substrates available for binding. And therefore you see the trend, right, that substrate concentration will reduce over time. Product concentration increases over time and then levels off when the reaction stops. Enzyme concentration, as you can see in this illustration, remains constant. There's like 7 of these. And they remain the same, they remain unchanged after a reaction. And we can see here that what is the limiting factor? Why can't the reaction goes on? In a time course reaction, enzyme concentration is the limiting factor. It is the one that stops the reaction, stops the rate from increasing further. So yeah, that's what happens. So if we take all these statements here that we just talked about and we plot them into graphs, it will look something like this. So here we have substrate concentration. We can see that it's high at first but decreases over time as it's being converted to product. So product, the line for product here over time is increasing because, you know, more and more substrates are being converted to product over time. Now you notice that it starts to level off right around here, and that's because the reaction has stopped. No more substrates left, therefore no more products to produce. Now the next interesting thing to notice is the enzyme line, so the dark blue. The enzyme concentration remains the same, you know, throughout the reaction because, well, again, it is not involved, it's not changed by the reaction. It is very involved in the reaction. It's just not changed. It's not used up in the reaction. It can be reused again and again. Now, the most fascinating thing out of this tree and the new thing here is actually the curve for the enzyme substrate complex concentration. So the ESC, or the enzyme substrate complex concentration, is actually really low at first and then increases steeply and then only decreases. Why is that? Why doesn't it just why didn't it start with high at first and then decrease over time? Well, that's because in this period of time, this very short period of time, there needs to be collisions that happen for enzymes and substrates to bind together. So it takes time for that to happen. It's not magic, okay? You don't put substrate and enzyme together and then it magically all magnetically attracts to each other and the reaction starts, right? Again, the substrate and the enzymes are randomly colliding in the reaction mixture, and it takes some time for it to collide in the right orientation and enough energy in order for enzyme substrate complex to form and a successful reaction to occur. Now, in all these situations are talking about concentration. Okay, but let's talk about rate a moment. At the beginning of the reaction, right, in this part, rate is high. There are more substrate present. There's a higher substrate concentration. But at the end of the reaction, rate is 0 because substrates have been all used up. So that's an important distinction to keep in mind. Are we talking about concentration of substrate or product or substrate complex, enzyme substrate complex, or is it enzymes? Like, is the question asking you about concentration, or is it asking you about rate over time? So yep, that's the time course graphs for you. Okay, so I know graphs can be confusing sometimes. They're a bunch of lines, but they mean so much. So I get this question very often, and I'll just like to start you off early with this. How to understand graphs? So the first thing here is always this. Yeah, you must always read the question carefully before you start to understand your graph. Understand what they are trying to say and trying to ask you. Are they asking you to describe, which is to state what you see, what you observe, describe a trend? Or are they asking you to explain why is it that it's decreasing? Don't tell me that it's decreasing, tell me why. Or are they asking you to compare and contrast? Are they saying, oh, can you compare and contrast between the concentration of substrate and concentration of product over time? That would really change how you analyze the graph. So always read the question carefully. The next thing you should always do is to read the axis labels. Now in this case, the x-axis is time and the y-axis is concentration. And it's very often, okay, actually always, this is always the case, the x-axis is always the independent variable. This is what you know as manipulated variable. But we don't use that word anymore. Take this word and throw it away. Yeah, no more of this word, okay? So the time is actually the independent variable, it's on the x-axis, and y-axis usually is the dependent variable. So in this case, it's concentration. So you are seeing how concentration change over time. Reading the axis labels is so important. There are so many graphs with the same shape, but different axis, which changes the meaning completely. So don't assume, read carefully and read the axis labels, and always remember x-axis is independent variable, y-axis shows you the dependent variable. Okay, that's how to understand it. How about talking about it? I see it, I see the trend. Where do I start? There's just so much data. Now, what they don't want you to do is to tell them each single detail about the graph. They only want a general trend. So describe to them how the independent variable and dependent variable changes overall. For example, in regards to substrate concentration over time, right? When substrate concentration, as time passes, substrate concentration increases, well, decreases, and then plateaus, right? So this is just an example. Maybe you can change it to product, it will fit, right? So when as time increases or as time passes, product concentration increases and then plateau. So this is a general trend. You don't need to tell them, oh, at 10 minutes, this happens. This is the concentration. At 20 minutes, this is the concentration. They don't want it. Just an overall general trend. Now, if needed, split the graph into several parts in order to describe and or explain separately, depending on what the question wants. So for example, we have, just now we said product concentration over time, right? So if the graph looks like this, okay, then maybe split it down to half and say that, okay, when at first, you know, in the beginning of time, when in the first part of reaction, what's happening to the product concentration, and then it plateaus, right? So what is happening there as well? Why is it plateauing? So that would be a very good structure to your answer. And the next thing that's needed here is a comparative data quote. Yes, they don't want you to quote every single point, but they want one quote, not to be lengthy, but to support your previous statement. For example, you said, okay, product concentration increases over time. So tell me, how does it, what points exactly show this? You must provide both the x and y coordinates with the correct units. So maybe this point here and this point here, you comparing between them and go like, oh, it has increased by how many, you know, moles, for example, the correct units over 10 minutes. So both the x and y coordinates, both concentration and time is needed as a comparative data quote in this example. Now, in some graphs, they have maximum and minimum value. So maybe a graph that goes like this, woo, right? So you can quote the maximum value if it's meaningful and appropriate. You can also include some manipulative figures, but I don't recommend this as much. For example, you can say that, okay, the number of cases reduced by half in 2010 compared to 2008. So you actually do some calculation and you come up with a smart way of saying things, but there's usually not a lot of marks for that. What you want is a general trend and a comparative data quote. Graph questions can go up to 4 or 5 marks per question, so make sure you know how to talk about your graphs. Don't worry just yet, you'll get a lot more practice as we go along. Just remember to do your homework and remember these 2 tips: general trend and comparative data quote. With that, we are done with our lecture. Remember to complete the assignment as proof of your attendance today. And yep, I will see you when I see you. Bye bye.